Good afternoon. Thank you all for joining our webinar. Today, we are pleased to introduce Dr. Michael Henry. Dr. Henry has been a faculty member of the Department of Molecular Physiology and Biophysics since 2004. The long-term research goals of Dr. Henry's laboratory are to understand the molecular and cellular basis of cancer progression and metastases in order to develop new methods for the diagnosis and treatments of the disease. Current efforts are focused on how cancer cells respond to hemodynamic forces while in the bloodstream. Dr. Henry also serves in senior leadership position as the Deputy Director for Research for the Holden Comprehensive Cancer Center. In this role, he seeks to develop the research capabilities and portfolio at the University of Iowa College of Medicine. And with that, Dr. Henry, over to you.
Thanks for the introduction, Melanie, and thank you all for coming. You've heard who I am. I have been a long-time user of bioluminescence imaging in our work around metastasis and using bioluminescence imaging to follow various therapeutic interventions. And my talk today is really going to focus on really how we've used this versatile technology in a variety of ways over the years. And I'll be illustrating some of this with the biology that we're particularly interested in. And I welcome your questions, either about the technology or even the biology that we're interested in at the end of my presentation. So we'll jump into it here. So this slide shows one way that optical imaging modalities have impacted cancer research over the years in terms of publications. You can see there's nearly 16,000 citations for optical imaging and cancer. This includes a variety of imaging modalities, optical imaging modalities, which all in one way or another use photons to construct images in living organisms. In the preclinical realm, bioluminescence imaging is limited to the preclinical realm because of the need to use a biologically encoded reporter system for it. Other modalities can be used preclinically and in clinical applications, and I've listed those there. I'm not going to mention any of those today. Really, our work has focused mostly on using bioluminescence imaging, but these also contribute to the publications over that time period. So I actually started using bioluminescence imaging round about the year 2000 when I was at Millennium Pharmaceuticals. At Millennium, we were an early adopter of the Xenogen IVUS technology. We evaluated this in my group there in cancer pharmacology and purchased this for use while I was there. And then I left Millennium and started a lab at the University of Iowa in 2004. And again, I purchased an IVUS 100 system when I started my lab here. And we used it until around 2012 when the cooling system failed for the last time. And since then, I've been using spectral instruments technology. In 2012, we began what was called then an AMI-X and what today is called the AMI-HTX system. And almost all of the work that I'm going to show you today is with the AMI-HTX system. All right, so many of you are probably familiar with what's encoded on this slide. The way non-invasive imaging modalities have been used in cancer research, but I'll just mention them once again. So this non-invasive imaging generally, not just bioluminescence imaging, but non-invasive imaging reduces animal usage by increasing the statistical power of studies through repeated measures. It enables humane endpoints based on disease burden. enables facile accurate quantitation of tumor growth, which in places other than the easily accessible subcutaneous space, which allows for more facile use of orthotopic and metastatic models. Also, importantly, this type of imaging is less prone to bias compared to traditional caliper-based measurements. It's more of a hard endpoint, at least in my opinion. Bioluminescence imaging and fluorescence imaging specifically enable the measurement of small number of cells, tumor cells. It's a very high sensitivity technique. It enables the use of sophisticated reporters for cancer biology, pharmacodynamics, and other purposes. And it's compatible with both transplant and autochthonous, which is a fancy word for genetically engineered models of disease. So I'm going to walk you through a series of just brief vignettes of some of the work that we've published since 2012 using the AIMI-HTX system. And the first one here involves a genetically engineered mouse model. In this case, we're using viral delivery of the Cre recombinase through the use of an adenoviral vector to different tissues to control tumor initiation. And in this particular example, we use it to generate a model of sarcoma. And so we're using a genetic background here that we had previously developed, where we have combined floxed alleles of the P10 and P53 tumor suppressor genes, along with a ROSA26 lock-stop-LOX reporter that allows for Cre-dependent expression of luciferase. And this is an allele that Bill Kalin's lab developed some time ago. So we've intercrossed these strains and back-crossed them extensively onto an albino black 6 mouse. These mice were all originally in the traditional black-coated C57 black 6 strain, but we crossed all of these mutations into the albino version of this strain because the lack of pigment in these animals facilitates bioluminescence imaging. The black pigment can attenuate the signal. So in this animal, wherever we inject Cre adenovirus, we can confer the recombination event that deletes two prominent tumor suppressor genes, P53 and P10, while activating luciferase expression. And so the next slide shows you how we've used this to build a model for sarcoma. So the site of tumor initiating cells for a number of sarcomas was unknown. And for this reason, we tried injecting the Cre virus into two different compartments, the subcutaneous space and the intramuscular space. And you can see from the panels on the right that both sites generated tumors that could be longitudinally followed over time. And that signal is quantitated in the panels below. Let me see if I can turn on my pointer and guide you around a little bit better. Okay. There we are. Okay, so yeah, so you can see in both cases, the subcutaneous and intramuscular injection, you get a rapid early growth phase of the tumor that then levels out into more steady growth over time. I just like to point out here, one of the things that we typically do when we analyze this data is that we will do a log transform of the data. So this is a log transform of the photon flux values measured from regions of interest that cover the entire animal in this case. And that's important for the statistical analysis when you observe events that occur over many orders of magnitude, like you see when you do bioluminescence imaging in mice. So that's one piece of the analysis that I like to emphasize. So one of the things that stuck out with these animals is that occasionally we would see some signal that was not in the region of the leg where we had done the initial injection. Here's a good example here from this mouse injected intramuscularly. We were seeing signal come up elsewhere. So we wouldn't have necessarily noticed this had we not been using bioluminescence imaging, but this clued us in to the fact that we were getting something that is not entirely unexpected. In other words, the virus was distributing systemically in this intramuscular injection, most prominently, and this was spreading to the liver, where it was resulting in recombination. And so when you do ex vivo imaging, you can see the signal showing up in the liver of the dissected livers. And when we did histologic analysis, you could see what the pathologist described as extramedullary hematopoiesis. This is not a liver tumor, but we were getting some recombination occurring in hematopoietic stem cells that were then proliferating in this tissue. We would not, again, I would say would not likely have been aware of this without doing bioluminescence imaging. So on the next slide, I wanted to point out something related to the sensitivity of bioluminescence imaging. So one way of measuring tumor growth in these animals after injection would be to wait until these tumors actually become palpable and measurable with a caliper. But that actually doesn't occur until about 10 weeks after the injection. Whereas with bioluminescence imaging, we can begin measuring tumor growth as soon as a week after injection and monitor this entire early phase of growth in these animals. And then at the end of the study, we removed these tumors and measured their volume and measured their mass and correlated this with, in this case, radiance measurements from the animals. And you can see there is a fairly good correlation for most of the tumors in this group of animals. However, there are some outliers, and it's important to know that bioluminescence imaging may not, in some cases, proportionally scale with tumor volume. And this, in our experience, this is mostly restricted to what happens when tumors get larger. And this can actually be a reflection of biology going on in the tumor. So when tumors get larger, they can generate necrotic regions, and those non-viable necrotic regions will not yield a bioluminescence signal, which is only apparent in the viable tissue. But this is actually useful to know, because very often what you're most concerned about is the viably growing tumor tissue and what you're doing to it, and less about the necrotic tissue. As the tumors get larger, there can be attenuation of some signal from the interior of the tumor. And that's related to those of you somewhat familiar with bioluminescence will be familiar with this problem illustrated over here, which is that essentially light is scattered and absorbed as it moves through tissues. And so it's very difficult, I'll say, in my opinion at least, to accurately reconstruct the volume of the absolute volume of tumors using just a standard bioluminescence imaging approach here. And so that light coming from the interior of large tumors can be attenuated somewhat, and then that affects the linearity of the signal. And then there can also be variations in how the tumor is exposed to luciferin, depending upon the vascularization of those tumors. So on to the next slide. Okay, here we are. All right, so on to the next model. Here in this paper, we've used the same genetic background that I just told you about, but instead of using adenoviral Cree, we have crossed those floxed P10 and P53 mice to a prostate-specific transgene, a probasin Cree animal, to drive expression in the prostatic epithelium instead of using that adenoviral technique. And this paper was actually a follow-up to a paper that we published in 2011 using the IVUS system, where we were just looking at the effect of loss of P10 alone in the mouse prostate. This newer paper, we use the AMI-HTX for imaging, and we're looking at the combined effect of loss of P10 and P53. So this shows the main finding of the study with respect to tumor growth. And what we could see is the addition of P53 here in red greatly accelerated the growth of these tumors. So when you have both P53 and P10 deleted, these tumors grew much faster. We actually expected this. Other groups had actually published this earlier, though not with the bioluminescence imaging angle to this, but it basically recapitulated some of these earlier findings. What is interesting, was interesting to me here, is that if we just look at the P10 alone mutants, which in this paper are these gray lines that extend out this way, they come up and then they flatten out and go along without a significant amount of change over a long period of time. This curve in great detail looks like the curve that we generated back in 2011 using the IVUS 100. It comes up at about the same time, levels off, and slowly increases. So, remarkably, using this mouse colony obviously was different after that long, and I had different people doing the work here, and the imaging devices were different, but we got a very consistent result with the P10 only group here using two different bioluminescence imaging systems. All right, this just shows some further characterization of this model, and this shows a technique that we and many other people have used a lot, which is often referred to as ex vivo imaging. So these are bioluminescence images that you take during a necropsy of a mouse, as you may also be aware, it's very difficult to use bioluminescence in this way to be quantitative to compare tumors. And the reason once you euthanize the animal, tissue begins to die. There's no longer a luciferin pumping around the system. and the signal can change in ways that are somewhat unpredictable. Plus, you're changing how the tissue is presented to the camera, and so we don't use it to quantitatively compare signal from one necropsied mouse to another. However, what we do use it for very effectively, I think, is to qualitatively assess where the signal is coming from, particularly when we're trying to assess metastatic disease, and then use that to focus our histologic efforts to confirm the signal. And so, what you see here, this is a body cavity of a mouse after necropsy and removal of the visceral organs. We often separate the organs out and look at them individually. But here you can see some signal that is we thought likely would be in lymph nodes. Again, this was a prostate model, so the prostate would be proximal to this lymph node, which would drain the prostate. And so we saw this signal and thought that it might be likely that this would be reflective of lymph node metastasis. But interestingly, when we dissected this tissue and looked at it histologically, what we found is that the tumor is actually not in the lymph node. The tumor is invaded a fat pad adjacent to the lymph node. So this is metastasis that has resulted, it's local metastasis that has resulted from extraprostatic extension of the tumor, not either likely from lymph node drainage of the tumor into the lymph node, or distant metastasis through the circulation, but rather by direct extension. And so that was the sort of interesting finding to us from that. And in fact, that actually jibed with others' interpretations of this combined P10, P53. They end up dying rapidly of very large primary prostate tumors and don't tend to generate distant metastasis. And I won't go through this, but we could see this in other visceral organs and in adjacent muscle tissue as well. So we also looked at castration resistance in this model. This is a typical question in prostate cancer: what happens when you withdraw androgens? And that earlier paper that I had mentioned, we found that when we castrated mice, there was some decrease in the size of the prostate briefly, but then it leveled off, and the disease never came back. Whereas in these P10, P53 mice, the tumor regressed, but then it came back with a vengeance in a castration-resistant state. So again, that was all generated with bioluminescence imaging. All right, on to a more recent paper we just published this year with our colleague here at Iowa, Don Quell. This was an effort to develop xenograft models of metastatic colonization for neuroendocrine tumors. And this is a type of tumor which, a type of model for these tumors that the field really lacked. And this is an area of interest of Don's. This example just shows some of the issues to think about when you develop new bioluminescent cell lines for tumor models in vivo. So we often now use lentiviral constructs to generate populations of cancer cells expressing luciferase and fluorescent proteins. And we tend to work with polyclonal cell lines. We don't select clones. But the lower example that's shown on this slide is the older days when we used to pick clones, and you can certainly still do that. Whether you generate polyclonal lines or pick clones, it's important to characterize these cell lines a bit before you go into an in vivo experiment. So in the top, this is from the paper with Dawn's group where we engineered the BON1 and QGP1 lines with a luciferase. In this case, we used nucleofection. We did not use lentiviral constructs, but we generated a polyclonal population anyway. And so it's important to test the overall signal level as well as its stability and uniformity before you go in vivo. And so one way to just test the level is to simply just do a limiting titration of cell number and measure the signal to get a representation of photons per second per cell. And in this case, we measured 30 photons per second per cell coming from the BON1 luciferase expressing cells, and a much higher level, almost 1,000 from the QGP1. So they can be quite different. In our experience, signals that are over 10 photons per second per cell are usually amenable to doing some of the metastatic models that we do. It's also important to test stability. And by stability, I mean what happens to the signal when you remove a drug selection marker that you use to generate the cell line. Does the signal fade away? Is the construct stable in those cells? So we often will pull the drug selection away for a matter of weeks and compare the signal of cells that are under selection and those for which we have removed the selection. And you can also test uniformity like we did here by plating the cells out at clonal densities and looking at the signals coming from individual clones. That will tell you in that polyclonal population how uniform is the signal. So in addition to these bioluminescence, the characterization of the bioluminescence properties, it's good to get some idea of the in vitro phenotypes before you go in vivo. In this case, Dawn's group looked at some cell migration phenotypes in the luciferase expressing cell line and the parental cell line, and these were quite comparable. And so that was reassuring that we haven't really dramatically modified the biological properties of the cells before we go in vivo. All right, so we have injected these cells in a couple of different ways. One of them is an intracardiac injection. Intracardiac injection, for those who aren't familiar, is injecting cells into the left ventricle of the mouse. And we've injected 1 × 10⁵ cells into the left ventricle. And this is in an NSG immunocompromised background. This intracardiac injection allows the cancer cells to pass through the arterial circulation and have access to certain organs in a way that they might not have access if you inject into the intravenous circulation. And what we saw here is in this intracardiac model, the QGP1 cells grew much faster. We've actually employed a clinical endpoint here of 10⁹ photons, so we can sacrifice mice once they pass this barrier, and allows us to euthanize mice with heavy disease burden without having to get them all the way to where they die in the cage. So it's a more humane endpoint. The tumor foci that we measured here by looking at individual signals was during that we looked at actually during a necropsy analysis. The BON1 cells had a greater number of tumor foci, so they were slower growing but had a greater number of colonies that showed up. Interestingly, the QGP1 cells showed greater colonization of the liver, which is very much a clinically relevant site of metastasis, whereas the BON1 cells showed greater lung colonization. That's also occurs clinically, but liver is more frequent. So we saw some differences then when we used an intravenous injection of the cells. Here we did 2 times 10 to the 5th cells IV into the same NSG host mice. We saw different growth rates here compared to the IC model. In fact, both models were much similar in this model, despite the big difference at the cellular level in the signal of bioluminescence that we see. QGP1 cells show even greater liver tropism in this model compared to the BON1 cells, which is kind of surprising because these cells have to pass through the lungs from a tail vein injection in order to get to the liver. And what this implies is that a lower inoculum of cells may reach the liver, but because QGP1 cells readily grow there, it eventually takes off in the liver. All right, so changing gears once again. This is some recent work from my lab related to our interest in how exposure to fluid shear stress affects the biology of circulating tumor cells. And here we were interested in the mechanism by which cancer cells avoid mechanical destruction in the circulation. And we discovered that this depends on a RhoA myosin 2 dependent mechanoadaptive process that occurs in cancer cells. And this paper itself highlights several different ways in which we've used bioluminescence imaging to explore this question. So just a little bit of background about this, our interest in this. This slide shows the trip that cancer cells take when they leave a primary tumor and enter the circulation. And so if you think about a prostate tumor as an example, they will leave this, enter the venous circulation in the pelvic floor, and go through an escalating level of fluid shear stress as they head toward the right heart. This journey takes seconds, and that's simply based on the time that it takes blood to flow from that region to the heart. From the right heart, they then go into the lungs, where they encounter a microvascular bed, the first microvascular bed that they see. And there are several fates there. Because cancer cells are relatively large compared to the microcirculation, they often readily lodge there, at least for periods of time. So that's what's shown here, the cells sort of being driven into this microvascular bed. And from there, some of them are displaced on into the other side of the circulation, where they'll enter the left heart. And then again, in a journey that lasts only seconds, they'll pass through the left heart into the periphery, where they'll encounter another microvascular bed. Some cells may extravasate and leave the circulation and potentially colonize the lung or liver, depending upon where the cells originate from. And some of those cells will die. And so we got interested in how cancer cells were experiencing the mechanical forces that they encounter along this journey. And there had been a prevailing idea that circulating tumor cells are inherently mechanically fragile and that they're destroyed by the forces in circulation. But there'd been actually very little evidence to support this idea. So we had a look at it, and we initially developed a very simple model, using a needle and syringe and a syringe pump to control flow rates through the syringe, to expose cancer cells to what amounts to a series of brief pulses of fluid shear stress. And this is, when I say brief, that we're talking millisecond duration pulses of fluid shear stress. That's the time at these high flow rates that cells are in the needle. And so we expose them to these repeated pulses of fluid shear stress. And then one way we've used bioluminescence imaging is we started using a lot of cell lines that were already engineered to express luciferase. And since we can use that as a measurement of cell viability, we see that over repeated pulses, you begin to lose viable cells. And that data is just shown here on the side. And the real upshot of this data, our basic finding, is that many different cancer cell lines are relatively resistant to fluid shear stress compared to non-transformed or primary epithelial cells. Blood cells, on the other hand, are much more resistant to fluid shear stress than cancer cells. You can see them here. So another way that we have used bioluminescence in this paper was to test whether cancer cells from real tumors are resistant to fluid shear stress in the way that we observed in cancer cell lines. And to do this, we took advantage of a mouse strain that I already told you about. This is mice with prostate tumors driven by probasin Cree, Rosa LOX stop LOX26, and then the P10 and P53 tumor suppressor gene mutants. And in this mouse, the cancer cells are labeled by luciferase, and we can isolate the prostates from these mice, get a single cell suspension, and then by following the luciferase expressing cells in an assay very similar to what I just described to you, we can measure the effect of fluid shear stress on viable cancer cells isolated acutely from fresh tumor tissue. And what we saw is that we saw a very similar profile to the cell lines that I showed you. In other words, these are prostatic cells from wild type mice. They're very sensitive to fluid shear stress. And those that have one or more of the tumor suppressor genes that are deleted show this resistance. So this experiment helped to show that fluid shear stress resistance is not just an artifact of cancer cell lines and is observable in primary tumor tissue. All right, so another way we've used it is to use bioluminescence imaging to monitor metastasis in mice injected with PC3 prostate cancer cells. And in this case, we had been learning about the mechanism, and we knew it was myosin 2 dependent. So here, what we did is we treated the cancer cells before we injected them into mice with non-toxic doses of blebistatin, which is a myosin 2 inhibitor. And then we injected them into the tail vein and looked at colonization days down the road or weeks down the road by bioluminescence imaging. And so we saw that those cells that had been treated with blebistatin before injection had a reduced, a delayed onset of metastasis, which is consistent with a reduction in the size of the inoculum because the cells were being destroyed while they were in circulation. There's other data that goes along to support this that I can't get into. And even though the effect is small here, because we're measuring this very sensitively and accurately, it's important to remember that we didn't treat these mice with blebistatin. The cells were treated with blebistatin before we injected. And so the time that the effect was manifest on the cells was very short, corresponding to the time that they were in circulation. All right. So another way we've used bioluminescence here is to take advantage of luciferase to measure the amount of cell destruction that take place immediately following IV injection of cancer cells into the circulation. In this case, we're measuring cell-free luciferase in the plasma compartment within a couple of minutes following injection of the cancer cells. And so, as you know, luciferase requires ATP and CoA inside of viable cells in order for there to be light emission. And when cancer cells are mechanically destroyed by fluid shear stress in the circulation, the luciferase is released from the cytoplasm and is present in the blood plasma briefly before it's cleared from the circulation. So by taking a plasma sample very shortly, and by very shortly, I mean within a few minutes of injecting the tumor cells, and then supplying ATP and CoA exogenously, we can measure the amount of luciferin released from destroyed cells. This is just showing this concept. So we can construct a standard curve from a known number of cells that we've lysed and then diluted into a blood sample, and then measure the luciferase activity, and then compare that with plasma that we've taken from mice and isolated. And so we can basically estimate the number of cells that are destroyed shortly after injection. And what we see is that, again, blebbistatin treatment increases the number of cells that are destroyed immediately following injection into the mice. All right, one last paper here is some work that we did with my colleague Chris Stipp, who is here at Iowa, on the role of alpha-3 integrin in prostate cancer metastasis. And so somewhat surprisingly found that a loss of alpha-3 function unleashed a much more aggressive metastatic phenotype in these mice, which we've measured in a cell line that expresses luciferase and enhanced green fluorescent protein. You can see the more rapid growth here in the alpha-3 knockdown cells. One thing to point out is that the intense signal that comes from the primary tumor, I don't know that I mentioned or made this clear. This is an orthotopic model. So we injected these cells directly into the prostate in this model and using a surgical intervention. And this intense signal from the primary tumor can obscure weaker signals that come from metastatic sites. And this can be revealed during necropsy, again, where you have the ability to look at individual organs. In this case, we use the fluorescence signal to evaluate that stereomicroscopically, because we were interested in really looking at microscopic disease that might be more difficult to detect from the camera on the AMI-HTX signal. So these are these stereomicroscopic images where we can then quantify the signal. The fluorescence signal you can more accurately quantify here because you're using fluorescent light to excite the fluorescent protein, and so that is more stable after you look in an ex vivo setting as compared to a bioluminescence imaging. We also used bioluminescence imaging to measure the growth, the three-dimensional growth of tumor spheroids in collagen gels. That can actually be a challenging setting to accurately quantitate the growth of spheroids. And here we can see, this is an experiment where we used an activator of ABL, which we found is actually downstream of alpha-3 beta-1 integrin in this the biology related to this model, and we can see that this DPH activator of ABL depresses three-dimensional growth, but has no effect on standard tumor growth. And here it was important to do a standard curve to compare the alpha-3 signal knockdown cells with the non-targeted controls to make sure that their signals were comparable. All right. Oh yes, one last, this is not published, but one last thing that we've done, and we've actually used our AMI-HTX to look at rat models of prostate cancer metastasis. And so this is our attempt to rescue an old sort of classic model of metastasis in the prostate field called the Dunning rat model. We actually couldn't get a hold of the Copenhagen strain of rat, which this was normally done in. So we've used immunocompromised nude rats in this case. This involves an orthotopic injection of prostate cells, 10 to the 5th cells. And these are, the cell line in this case is this Matli-Lu, in which we've engineered with luciferase and GFP in similar manner as I told you about before. This is a very aggressive model, very rapidly grows. The primary tumors get massive. You can see that they're within less than 3 weeks, we have tumors that are around 10,000 cubic millimeters. So they get they grow rapidly, very, very fast. And then we can do necropsies on these to further characterize the metastatic phenotype. And we see this evident in necropsy, again, in this case, in liver and kidneys. We can also flush bone marrow from these mice, and we can see GFP positive cells in the bone marrow. And we can also measure GFP positive circulating tumor cells from blood samples. All right, we've used the technique in a variety of other papers that we published in the last almost 10 years since we've been using the AIMI-HTX system. I'll just flash a few of these up here, doing different things. And then I'll just end up here with a few words about the system itself. So back in 2012, when we were considering replacing our IVUS 100, we had some questions about the relative sensitivity of what was called the AIMI-X at that time in the IVUS system. So we had a look at this using a calibrated light source, where we compared our IVUS 100 and our core here at Iowa actually had an IVUS 200 system. So we were able to look at that as well as the AIMI-X. And really all three of these systems were within 30% of the calibrated value, with the AIMI-X and IVUS 100 being the closest. And so in terms of sensitivity, we also compared IVUS 100 and AIMI-X in what is a high sensitivity application. In this case, we used that viral Cree model that I told you about at the very beginning. Here we injected it into the tongue of the animal. I had a postdoc who was interested in trying to develop a model of oral squamous cell carcinoma, and ultimately that failed. But what the bioluminescence imaging did allow us to do is to confirm that we were getting recombination of the luciferase. These mice weren't developing tumors, but we were seeing evidence for recombination. And this is, the signal here is really right down near the background levels that you get, down around a few thousand, radiance levels down around a few thousand photons per second per centimeter squared per steradian. So, and you can see we got comparable signals on both systems. And then with some colleagues at Spectral Instruments, we had a look at another thing. So when you're imaging down at these very low levels, the lens performance actually becomes more of a significant issue. So along with Mike Cable, who was at Spectral Instruments at that time, we used, we took a look at these lens performance issues. So we also use this high sensitivity imaging to assess injection success in mice that have been injected intracardially. So when you see signal of injection of this relatively small number of cancer cells systemically into the mouse, and you see signal that's concentrated in the thoracic cavity, that is indicative of at least a partially failed injection. Whereas injections where you see no signal and the cells are fully distributed systemically, you don't see any signal. And to do this, you really need to use high sensitivity imaging. And on the IVUS 100 system, what you see is this halo effect where you see this signal all around the edge, and that's evident in this line scan across the signal. And essentially what's going on here is there's an algorithm that attempts to correct for the lens performance in what's called a flat fielding algorithm. And so what it eventually does is it can amplify noise around the edge that you only see when you're doing high sensitivity imaging. So we took a calibrated light source and measured this at the center and at the periphery of the signal. And the data is quantified down here. And we measure this with the flat fielding systems on and off. And the flat fielding algorithm in the IVUS 100 produces a greater effect. In other words, it's compensating more for the lower performance of the lens in the IVUS 100. The AMI X or HDX is actually comparable to the IVUS 200 system that you can see here in the data. So this really is only a consideration when you're trying to look in the most high sensitivity environments. And essentially what this shows is that the lens performance on the AMI HTX was comparable to the much more expensive IVUS 200 system. That's that. I'd just like to thank folks in my lab over the years that have contributed to the work that I've showed you, as well as collaborators here at Iowa and other institutions, and funding from these various sources to make this possible.